Good morning, everyone. Greetings of the day. I, Professor Dr. Simajit Shergill, welcome all of you in the third and the final day of the workshop on Life Science, Food Science and Environmental Technology 2021. I welcome our worthy Vice Chancellor, Professor Dr. Parvinder Singh, respected Registrar, Deputy Registrar, Deans, Heads, faculty members, dear students and participants. I am very thankful to our speakers of this session, Dr. Sonia Wycombe and Dr. Ram Avatar. And I'm extremely thankful to all of you for your keen interest and active participation in the workshop for the last 2 days, and very much overwhelmed to receive a positive feedback from the participants. So once again, I welcome all of you in the third and the final day of the workshop. Now, I would like to give a brief about the today's session. Our first speaker of the day is Dr. Sonia Wycombe from the Manipur University, and she will speak about the various gene expression techniques. Our second speaker of the day is Dr. Ram Avatar, a scientist from Institutes of Biotechnology, Delhi, and he will enlighten our participants about the genome sequencing, assembly, and the annotation. So afterwards, we will be having the concluding remarks of the 3 days workshop and followed by the vote of these thanks. So once again, on behalf of Ratan Bhara University, I welcome all of you. Now, I would like to introduce our first speaker of this session, Dr. Sonia Wycombe. Currently, she is in the Department of Life Sciences at Manipur University, and she got her PhD from the Department of Botany, University of Delhi, and she worked upon the development of the transgenic plants using lactazines from onion and the garlic for the introduction of the aphid resistance in the oilseed crop. She worked as a research associate in the project entitled Clonal Propagation of the Elite Genotype of the Orchid synthesized in the Manipur for the commercial cultivation. Her thrust area of the research, if I say, are the plant biotechnology and the molecular biology, total DNA, RNA isolation, purification and the quantification, PCR and the gel electrophoresis, preparation of the competent cells, cloning methodology, plant tissue culture, and bioinformatics tools. She has presented her research work at the various national and the international platforms and has published many international research publications in the research journals of the high repute. Now I invite and request Dr. Sonia Wycombe for enlightening our participants about the various gene expression techniques. Dr. Sonia, over to you.
Thank you, Dr. Samarjit, for having me. Such a pleasure to be here talking, giving a presentation here at your university. I'm very extremely sorry that you won't be able to see my video. There's some technical issue. So I'll be just, you will be just hearing my voice. Unfortunately, you'll not be able to see me because there's some technical issue with my computer. So without any further ado, I want to start my presentation. So just a second. So today's topic of my talk will be gene expression analysis. There are various techniques available which are used for expression of genes. So we will be discussing or talking about some of the techniques which are being routinely used to check the expression of genes. So before before talking about the gene expression techniques, let us first know what is gene expression analysis. Gene expression analysis is nothing but the study of the way genes are transcribed to synthesize functional products such as functional RNA species or protein products. Now, this gene expression analysis, it gives an idea about the normal cellular process, such as differentiation in cells, and it also helps us to, compare the expression of gene levels in abnormal and pathological plants or cells or tissues. So in that way, if we know which gene is expressed in a disease cell and which is not expressed in a normal cell, it can help in the detection of the disease and also in the development of the drugs to cure that particular disease. So expression analysis can be done at various levels where gene expression is regulated. Example, at the transcription level, at the post-transcription level, translational level, and the post-translational and protein modification level. Okay, so what is gene expression? So let's first give a brief idea about what is gene expression. Uh, gene, ex Gene expression is the transcription of the RNA into a DNA copy, which ultimately then gets translated into protein. This is how a gene is expressed. So this expression of gene can be regulated in a number of ways. So one of the most important way of gene regulation is the binding of the regulatory proteins to DNA binding sites. So where the DNA regulatory proteins binds to a particular DNA site, that gene will be expressed. And if those regulatory proteins are repressed or not present, that particular gene will not be expressed. And the other a very important way of gene expression is again methylation, DNA methylation. Sorry for this. This should be DNA methylation. DNA methylation on gene expression. We know that the genes which are highly methylated, they are not expressed. They are repressed. They are and those genes which are not methylated or which are acetylated, gene expression, gene regulation. And gene can also be regulated after transcription, that is at the post-transcription level, depending on the stability of the RNA. And it can also be regulated at the protein at the translational level, where the protein gets degraded, or the stability of the protein also defines the regulation of a particular gene. And the conformation of the protein also can dictate the function of a gene. So it can also be regulated at the post modification of protein also. Now, this is a brief idea of how gene is expressed, how it is regulated. Now, how do we check this expression of gene and gene? Here in this presentation, I'll be talking about some of the techniques which are available for checking the expression of the gene. And a very important and a very routinely used method of to check gene expression is to check the expression of RNA, because RNA is the first biomolecule which is formed after DNA. So some of the routinely used RNA expression techniques are Northern blotting, DNA microarrays, and real-time PCR. In this talk, I will be covering in detail DNA microarrays and real-time PCR. And I'll give a brief overview of the other techniques also, but the DNA microarrays and real-time PCR I'll be covering in detail. So let's see what is the first RNA expression detection technique, that is the Northern blotting. It is nothing but it is used to check the presence of RNA in a particular sample. What we do in Northern blotting is we extract the RNA from a sample, from our desired sample, then we run it on an electrophoretic gel. After the sample is run on an electrophoretic gel, it is transferred into a probe, and the probe transferred into a blot. The blot is then incubated with the specific probes, to detect the presence or absence of our gene of interest. If there is hybridization or if there is a signal, then it dictates that there is the presence or the expression of our gene of interest. And the second method is the DNA microarray. DNA microarray is nothing but an array of oligonucleotide probes bound to a chip surface, which enables us to detect the expression profiling of many genes in response to a condition. This oligonucleotide probes and chip surface, this I'll be covering in detail. So I'll show a picture of all the probes and the chip surface also. Then the next, which is very, very much in use during this pandemic, is the real-time PCR. It is the steady state level of mRNA are quantified by reverse transcription of the RNA to cDNA, followed by the quantitative qPCR on the cDNA. Now, we must have all be hearing about RT-PCR, the detection of coronavirus, because this has been routinely and is the gold standard detection technique for coronavirus. So we'll be covering in detail this RT, real-time PCR. In this real-time PCR, we get to know the quantity and the quality of the RNA expressed in a particular sample. After talking about the RNA expression technique, detection techniques, let's talk about some protein expression analysis technique. The first one is Western blotting. It is very commonly used technique for protein expression detection. It is similar to Northern blotting. It is just that instead of RNA, we use protein for quantification. We run a gel after extraction of the protein. We run a gel, then the gel is transferred to a blot, then again incubated with probes to check the expression of our protein of interest. Then the next is the 2-gel electrophoresis. Here, the protein expression profiling is achieved by separating a complex mixture of proteins into 2 dimensions and then staining to detect differences at the whole proteome level. The third technique is the immunoassay. Proteins are quantitated in solution using antibodies that are bound to color-coded beads. As I have said in my earlier slides also, that proteins can be regulated at a post-modification level also. And here I have covered 2 techniques which can be used for post-translational modification analysis. The first one is the immunoassay. Here, the levels of protein phosphorylation and other translational modifications are detected using antibodies that are specific to these adducts. And the last one is the mass spectrometry. Here, the protein and their modifications are identified based on the differences in their masses. I have given a brief overview of the various techniques which I have used for detection of expression of RNA and proteins and post-modification of proteins. Next, in this slide, I'll be covering in detail DNA microarray technique. As I have said in earlier, the DNA microarray is a tool to detect the expression of thousands of genes at a time. So we can check the expression of many genes at a single go. So it is a very useful technique. Here in the DNA microarray, we use microscopic slides, which I'll show you in the later slides. And in this microscopic slides, there are thousands of tiny spots present. And in these tiny spots, to these tiny spots are attached the known DNA sequence or gene sequences. In each of the spot, a DNA sequence or a gene sequence is present for which the sequence is already known. Then we isolate RNA from our sample, from our interest sample. Then those RNAs are labeled, and these RNA are then hybridized to the tiny spots of the microscopic slides. If the DNA sequence if our gene of interest is present in the slide, in the microscopic slide, it will hybridize and it will show a signal. And showing a signal means that that particular gene is being expressed, because we use different color codes, and the different color codes will tell us which gene is expressed and which is not expressed. Now let us look at some of the principles which is followed in the DNA microarray technique. I'll just give a brief overview of it. We know that the principle of DNA microarray lies on the hybridization between the nucleic acid end. That is the nucleic acid of our sample and the nucleic acid present on the microscopic slide. And the property of complementary nucleic acid sequence is to specifically pair with each other by forming hydrogen bonds between complementary and nucleotide base pair. We all know that the 2 strands of DNA are bound together by hydrogen bonds. Then for this, the samples are labeled using fluorescent dyes. Different dyes will be used to detect the presence of different genes. And at least 2 samples are hybridized to the chip. For example, we can take 2 samples from a disease cell and a normal cell, or from 2 tissues of the same organism. Complementary nucleic acid sequence between the sample and the probe attached on the chip get paired via hydrogen bonds. After the non-specific bonding sequences will remain unattached and wash out during the washing step, all the sequences which are not bound to the microscopic slide will be washed away. The fluorescent label target sequence that bind to a probe sequence generate a signal. There will be a laser detector. It will detect the presence or absence of a signal. The signal depends on the hybridization condition, that is the temperature, washing after hybridization, etc. What it also strength of the signal depends upon the amount of target sample present. Using this technology, the presence of one genomic or cDNA sequence in many sequences can be screened in a single hybridization. Now, after going through the principle, let us see the process or the protocol of DNA microarray. The reaction procedure takes place in such a way. First, we collect the samples of a normal cell. As I've said earlier, we can collect the sample of a normal tissue and or a diseased tissue. We collect, and the sample should be homogeneous. It should not be very heterogeneous sample. Then, after collection of the sample, we isolate the mRNA. mRNA can be extracted using kits which are available in the market, or we can use the simple solvents present in the lab, like using phenol chloroform. And when the total RNA is extracted, we will separate the mRNA from the rest of the RNA, that is rRNA and the tRNA. This mRNA separation can be done using column beads, which has poly T tails. We know that mRNA has poly A tail. This poly A tail will hybridize with the poly T tail, and the then after the extraction of the mRNA, we need to convert the mRNA into cDNA. Because we know the DNA is more stable stable than RNA, we have to convert into cDNA so that we can utilize the cDNA for further reactions. So to create the cDNA, we use reverse transcriptase enzyme. There are kits available to convert the cDNA into mRNA, simple kits which they give along with the enzyme, and we can convert it in a simple PCR machine. Then after converting the mRNA into cDNA, we incorporate the samples with the fluorescent dyes for producing fluorescent cDNA. This will help in distinguishing the kind of the DNA of the gene which is expressed. Then the next step is hybridization. The labeled cDNA from both the samples are placed in the DNA microarray so that each cDNA gets hybridized to its complementary strand. They also are thoroughly washed to remove any unbound sequences. The last step is the collection and analysis step. The collection of data is done by using a microarray scanner. The scanner has a laser, a computer, and a camera. The laser will excite the fluorescent in the labeled cDNA, and it will create a signal. When the laser scans the array, the camera records the images produced. When the computer stores the data and provides a result immediately, the data will then be analyzed. The difference in the intensity of the colors will also give an idea about the amount of the hybridized DNA samples. Now this slide summarizes what I have talked in all the summarizes the experiment of the DNA microarray. So here we take a normal sample and a cell and a cancer cell. This is a disease cell and a normal cell. And from these two samples, we isolate their mRNA, RNA isolation. Then this RNA is then converted into cDNA. Then after conversion of the cDNA, they are labeled. For example, in the normal cell, we use green fluorescent probes. So the cDNA sample for normal cell will be labeled green, and the cDNA from the cancer cell will be labeled red. This is the microchip or the microarray chip that I talked about. There are many slots present. These are the small slots present. And in each of the slots, there is a DNA sequence or a gene sequence of a known sequence. The gene sequence will be known, or if you use an oligonucleotide, that sequence will be known. So you know that which particular gene is present in each of these slots. So then after the cDNA is labeled, you put these samples in each of the slot, okay, in each of the slot. And if this, for example, if a gene from this normal cell is present in this slot, it will hybridize. It will hybridize, and when you scan it, it will give a signal. For example, say in this particular slot, yellow color is present. What does it mean? What does yellow color indicate? We know that green and red, when they combine together, it will give a yellow color. yellow spot, it means that both genes from both the normal and the cancer cell have hybridized to the DNA sequence present in this slot. That it means that both these genes are present in this, are expressed in both the cells. Whereas if in a particular slot only green signal is present, it means that the mRNA from the normal cell have hybridized to this slot, and this only the genes from the normal cells are expressed in this particular gene, in this particular slot. For example, let's say that the uh, for example, in this slot, red signal is present. What does it say? What does red signal in this slot say? It indicates that the mRNA from the cancer cell is hybridized to this red slot, and it indicates the expression of that particular gene in the cancer cells. So using the scanner and the different color which in both the cells. So after understanding the principle and the process of the cDNA, let's see what are the applications of DNA microarray. It can be used to determine how particular disease affect the pattern of gene expression in various tissues. It also helps in discovery of drugs, diagnostic and genetic engineering, alternative splicing detection, proteomics, functional genomics, DNA sequencing, gene expression profiling, and toxicological research. Now let's look at some of the advantages and disadvantages of DNA microarray. Advantages of DNA microarray are it provides data for thousands of genes in real time. The expression of thousands of genes can be checked in a single experiment. Single experiment generates many results easily, fast and easy to obtain results. You can just check the colors which are being present, and you can tell which gene is expressed and which is not. And it is promising for discovering cures to disease and cancer, and different parts of DNA can be used to study gene expression. And some of the disadvantages are it is expensive to create, the production of too many results at a time requires long time for analysis, and the DNA chips do not have a very do not have a very long shelf life. So here with this slide, I will end the topic on DNA array. Next I'll be covering the real-time PCR. Real-time literally means in the actual time. So what does real-time PCR means? Real-time PCR means that it measures the amount of PCR product at the end of each amplification cycle. For example, in conventional PCR, if we run a PCR, we have to run the DNA products or the amplicon and on a gel to see the result. But in this, we don't need to run on an electrophoretic gel. The machine of the PCR will indicate if the gene is expressed or if the gene has or tags to label the PCR products. So when the PCR products are labeled, it will indicate how much DNA has been amplified or how much amplicon is present in the reaction. The main advantage of the real-time PCR over PCR is that the real-time PCR allows to determine the initial number of copies of template DNA. We can make a comparison of two samples and check in which sample initial DNA is more or less. Real-time qPCR can be evaluated without gel electrophoresis. This I've already told, resulting in reduced bench time and reduced throughput. Now let us see how does real-time PCR work. Here also we collect the sample. The sample should be homogeneous. Then the next step is after collection of the sample, we extract the DNA. We can use the same technique as we have done for DNA microarray. Then we analyze the nucleic acid quality. The RNA concentration is analyzed using a spectrophotometer at UV absorbance of 260 nanometer or 280 nanometer. Then after the quality check, then we transcribe the RNA into cDNA. It's the same process as we have done in DNA microarray. It's just that in DNA, real-time PCR, we will conduct a PCR of the cDNA. In real-time PCR, detection system consists of a thermocycler. This thermocycler is equipped with an optical detection module, which will measure the fluorescence signal generated during each amplification cycle as the fluorophore binds to the target sequence. Now, in real-time PCR, the detection can be done in two ways. One is in a very specific way, and another is a non-specific way. In a non-specific detection, we use a dye that is a cyber green. And I'll tell how it is different. And in a specific detection, the fluorescent reporter probe will be used. Example is a Tagman assay. Now let us first see how cyber green detection is done in real-time PCR. Okay, when we run the PCR using the cDNA, we add all the components of the PCR, that is the polymerase enzyme, the DNTPs, the primers and everything. Along with that, we will add the dye, that is a cyber green dye. This cyber green dye is binds to double strand DNA. Okay, it binds to double strand DNA molecule. So when we start the reaction, the number of the amplification, depending on the amplification cycle, the DNA amplicon will increase, the DNA products will increase, the number of the double strand DNA will increase. So as I have said, this cyber green binds to double strand DNA. So with the increased number of the amplicon, with the increased number of double strand DNA, there will be an increased binding of the cyber green dye on the double strand DNA. So the intensity of the fluorescence will increase along with the increase in the number of the amplicons and the number of the PCR cycle increases. And when the amplicon and when the signal has reached a threshold level, then the scanner will detect the signal and it will tell us that this much cycle has proceeded, and it will also indicate how much of the amplicon is present. So this is a detection method of non-specific detection method using cyber green. Next is the specific method using this is common routinely known as a Tagman assay. In this Tagman assay, we use a probe instead of the dye. We use a probe. This probe has a fluorophore. This R is the fluorophore. This and Q is the quencher. What happens to this probe is when the quencher and the fluorophore are together, it will not emit any signal, because the signal which is emitted by the fluorophore will be absorbed by this quencher. As long as they are together, the fluorophore will not be able to emit any signal because the signal is being quenched by this quencher. Okay, so when we start the reaction, we add the probe, the primers, and all the other components of the PCR reaction. And when we run the cycle, we also add Tag polymerase in the enzyme for PCR amplification, right? So this DNA polymerase, Tag polymerase, it has a 5' exonuclease activity. What happens when we run the cycle, when the primer has bound and the DNA polymerase starts amplifying the DNA, it encounters the probe here. So since the Tag polymerase has exonuclease activity, it will cleave the fluorophore from the quencher. So once the fluorophore is free from the quencher, the fluorophore will be able to emit the signal. Since it is not together with the quencher, the signal will not be absorbed, and we can see the signal. So the signal will be visible and can be detected by the scanner. So when the reaction cycle increases, more probes will bind to the DNA sample, and the Tag polymerase will cleave more of the probes, and more signal will be emitted, which will be captured by the scanner. Now this is a graph which shows the real-time PCR reaction. Here on the y-axis, it indicates the fluorescence of the DNA product, and on the x-axis is the number of cycles. Here we saw a green line here, and there is a threshold line. This threshold line is nothing but it signals the presence of a of the fluorophore. The fluorophore will emit the light when this particular cycle has reached. For example, in this graph, we see that at around the cycle number 20, the CT value is rich. CT value is threshold cycle, the cycle from which we will be able to see the fluorescence. So when this in this sample, when the PCR cycle has reached 20, the amplicon will be able to emit the signal, and the signal will be captured by the machine. This is, and we can also compare two samples. For example, if there's a sample A and it fluoresces at 20 cycle, and if there is a sample B, if it fluoresces at cycle 30, it means that it is taking higher number of cycles to emit the fluorescence. So we can make a comparison of how much DNA is present in the sample in the initial cycle. For example, in this green graph, if the sample is will have higher number of template as compared to a sample which fluoresces at cycle number 30. This is all for real-time PCR. Now let us look at some of the applications of real-time PCR. It is a related and absolute quantification of gene expression. It is used for validation of DNA microarray and variation analysis, including SNP, discovery and validation, counting bacterial, viral, or fungal loads. As I've said, it is routinely being used to detect the coronavirus. Thank you. That's it.
Thank you so very much, Dr. Sonia. You can stop sharing your screen.
Okay.
So thank you so very much, Dr. Sonia. I'm highly grateful to you. You delivered a very informative talk on gene expression, profiling, and quantitation, and explaining the various gene expression techniques. So this was very valuable session for our participants. Though due to some technical issues, we were not able to see you, but your visibility was there throughout this speech with your words. A very impressive speech. So thank you very much. Now this session is open for the students. If they have any queries, then afterwards we can propose a vote of thanks. Participant, if there are any queries, or we can proceed ahead. Yes, Dr. Sonia, one of our participants is asking which detection method is better for RT-PCR.
It depends on our experiment, on the kind of experiment that we are doing. For example, CyberGreen is very cheap compared to Tagman. Tagman is again expensive. So if you want to detect the expression of gene, and if it is not very specific, you can go with CyberGreen. But if you want a more specific detection of the gene, then you have to go with Tagman Assay. So it depends on the experiment.
Right. So thank you so very much for your answer. Now I invite Dr. Vandana Patyal to propose vote of thanks for the speaker of the session. Dr. Vandana Patyal, over to you.
Thank you, ma'am. A very good morning to all. I'm Dr. Vandana Patyal, Assistant Professor in Life Sciences. On the behalf of Raita and Bahara University, would like to thank our Honorable Speaker, Dr. Sonia Wycombe, for enlightening us about different techniques of gene expression, like different blotting techniques, northern, southern, and western, RT-PCR method, DNA microarray, and their application in medical field. Right. So, Dr. Sonia, this information will definitely help our students, especially with biotechnological subjects. And once again, I would like to thank Dr. Sonia for sparing some time from her busy schedule. Thanks, Dr. Sonia. Now, I would like to invite Professor Simarjit Kaur Shergill to carry on with the next session. Thank you, ma'am.
Thank you, Dr. Vandana, and thank you, Dr. Sonia, once again. Thank you.
Thank you for having me. It was a great pleasure.
So I can see our next speaker of the session, Dr. Ram Avatar. A very heartiest welcome to you, sir.
Thank you.
Now, I would like to give a brief introduction about our second speaker of this session, Dr. Ram Avatar. He's currently working as a research scientist at ICR National Institute of Biotechnology, Pusa campus, New Delhi. He did his PhD from Research School of Biology, the Australian National University, Canberra, Australia. And he is recognized with various awards and the scholarships. He got a scholarship from Department of Biotechnology during MSc, then ICR Senior Research Fellowship, then CSIR Junior Research Fellowship, and ICR International PhD Fellowship for PhD at ANU Canberra. So we can see he got himself entitled for all the fellowships and the scholarships. And he has been participated in the various, national and the international workshops, and having, international publications in the research journals of high repute. And his research work is speaking in the form of his research publications. And very, very good publications, sir. I was just going through your, the brief profile, and I'm really amazed to see your, the achievements. And Dr. Ram Avatar is, he has worked on various projects, and few of which I am going to mention right now. He worked on a project entitled Molecular Analysis of the Coat Protein Gene of Lily Symptomless Virus Infecting Lily and the Tulip. He also worked on a project entitled Development of Zinc and Iron Enriched Maize through Molecular Breeding. He worked on a project entitled Identification of Functional Relations between microRNA arising from a large genomic cluster. And he also worked on computational and the functional analysis of the defense response genes involved in the sheath blight resistance in rice. So if I will keep on talking about the projects he has worked on, it is going to be a very, the long list of the projects he has handled. So thank you so very much, sir. We are really enlightened to have you over here today. Now I invite you to enlighten our participants with your expert talk. Over to you, sir.
Thank you, Professor, for generous introduction. I'm going to share my slide now. Can you see the slides?
Yes, sir, we can see it.
Am I audible?
Yes, sir. You are audible as well as visible. Thank you, sir.
Okay. Thanks again, Professor, for the invitations, and good morning to all the participants. I'm going to discuss a little bit about bioinformatics, as I'm a biologist turned bioinformatician, and during my PhD, my training was most of my training was on hardcore bioinformatics. So I'll touch upon some concept of bioinformatics, and which has become very important in this day and age. It's become a very integral part of biology. You can't do good biology without help of bioinformatics. So what I do, I'm a fungal molecular pathologist. What I try to do, I do genome sequencing, take the help of genome and see the interaction happening between the fungal pathogen and plants. To understand the interactions and then try to make plant more resistant and tolerant from fungal insects. So, okay, let's.
Dr. Ram Avatar, I guess your voice is cracking in between. Maybe you have put the headphone only in your one ear.
Okay, sure. Am I clear now?
Yes. Okay. Thank you so much. Thank you.
Yeah. So before we go into dwell into genome sequencing, we have to understand what is genome is. And most of you are already familiar with the genome, and you might have read about genomics and during your undergraduate course, or you might be taking some courses in your current course curriculum. So genome is like the term genome came in 1920, where a German botanist gave a term genome, where he defined genome as a haploid chromosome set together with the pertinent protoplast, which specific the maternal foundation of the species. It only mean that like your haplom is like one half of your chromosome, either coming from maternal or paternal, is your genome, which contain all the genes which make a functional copy of an organism, like one functional copy of human, plant, insect, for that matter, and fungal species. So, but that terminology came in 1920, but it has been evolving concept. So genome is just not about the information coded into nucleic acid, but it is, you might have heard of technology like terminology like epigenetics, which is beyond the DNA sequence. Epigenetics is information dealt with the methylation of the base pairs and also methylation of histone molecules. So that again put another layer of regulation, genome regulations and regulation, and it also helped in shaping an organism to an ecological niche. So this is a Google analytical search of from genome, chromosome, and genes. So you would see like until 1960, from the date of the term came to existence 1920 to 1960-70, there was no use of it much. But after DNA discovery in 1953 by Watson Crick, it has started being using slowly into the books and research articles. But it use has become exponential after coming genome sequencing technology. Since the DNA discovery, people have been trying to put together whole genome of many organism to understand what is in the genome. So and now after next generation and now third generation sequencing technology, the genome sequencing is become just a couple of days job now. So it is going to be used even more in days to come. So this is a representation of a whole genome of rice into 24 chromosomes. So as I was explaining, like what is genome is, like you have 24 homologous chromosomes. One chromosome copy comes from the maternal, inherited from maternal, and one from paternals. So one of these copies put together 12 chromosomes makes a rice genome. So rice genome, you look at the chromosome level, looks very compact. But if you start unwinding chromosome, you would see DNA strain around the nucleome stacked together in a chromatid, which is the smallest constituent of chromosomes. But if you unwind it further, what you end up getting the DNA sequences. So what DNA sequencing try to aim is like fragment these, extract DNA, fragment them, and sequence and try to assemble them in a one sequence after another on the basis of DNA homology. And every genome sequencing aim to produce a read sequence of a chromosome from one telomere to another telomeres. But this is the ultimate aim. But getting to this feat of getting a chromosome, a DNA sequence from one telomere to another telomere is a really daunting task, and a lot of technology is required in order to achieve here. Still, there has been a lot of evolution in the genome sequencing, started from the dideoxygenome termination sequencing invented by Sanger to now third generation sequencing, where you use PacBio and nanopore technology, where your average read length is 10 kb. Still, it's difficult to achieve because these chromosomal telomeric and centromeric sequences are still difficult to assemble. So why do we sequence a genome? So obviously, like being a biology student, you must be asking why do we have to do a genome sequence? So as I explained, like all the genes are are in the genome, and the genes makes you what you are. So any biology, if you want to understand any biological organism, you have to see the DNA and nucleotide level. What is it constitute of? What kind of gene it has? What kind of transcription factor it has, and how they express in different tissue at the different time, spatial and temporal way. So that information you could only get once you have the entire genome sequence of an organism. So that is the purpose of genome sequencing. Yeah, another point to highlight that whole classical taxonomy has been now pretty much replaced by the molecular evolution by using understanding relationship between two organisms on the basis of DNA rather than the common feature they share, because those feature can be misleading, as you might have heard of the terminology of convergent evolutions, where organisms would not have any correlation at the genome sequence level, but they have similar features because they happens to evolve in a particular common environmental niche. So genome size conundrum, you might have already have heard of it. The genome size does not correspond to the complexity of the organism. So similarly, you see there's a whole lot of variation in the genome size. Wheat genome, which is like one of the biggest in a cultivated agricultural crop, which is as big as like almost 5 times the genome size of human. It's such a complex genome because it's a polyploid genome constituted of 3 different genomes. And then simplest one is the Arabidopsis with just 120 MB millibase pair, and it has been sequenced very well, and it has been a model organism to understand all other biological concept in biology. So I've already discussed about the evolution of the genome sequencing, but just to go again over it. So 1970, Mischer discovered DNA. Then the 1940 53 Boston Crick described the DNA double helix, and from there onwards, it has been like a steady state progress in understanding and trying to sequence the genome of a whole organism. And then Sanger sequencing came in 1970, and now the things has gone exponentially, like you producing DNA sequence in an exponential way. Sequencing genome organism of as complex as a human, which took 10 years in a first attempt to sequence genome, started in 1990 and took almost 10 years to finish. But now a human genome can be sequenced and assembled in just a week time with the very, very minimum resources of money required. So these are the sequencing technology currently used, and some of them has been like outdated, like the ABA 454 sequencing came in a early 2000. But now most of the sequencing technology are based on Illumina, where they produce short sequence but high quality sequences, very high proofreading. Then those sequencing, these Illumina sequencing technology have drawback this because of their short size. They are not very quite helpful in the genome assembly, because the eukaryotic genome are complex, full of repeats, and those repeats could not be spent through by these short reads, and that create a lot of complexity and produce a highly fragmented genome. So in order to achieve a better contiguity genome, contiguity to the point of like as I explained earlier, that contiguity has to be at the level of like chromosome. Like you start from the 1 telomere of the chromosome and you end up another end of the chromosome. That level of the contiguity is strived and scientific community is striving to achieve someday. It has been largely achieved with the help of third generation sequencing technologies such as PacBio, MinION, Oxford nanopore sequencing technology, where the average read size is about 10 kb. Just imagine like this. Sanger sequencing technology could produce a read from both ends 700 base pair at the most, but now its average read size can be achieved to 20 to 50 kb after a proper size selection. Again, this is again a very classical diagram to just show the how sequencing evolved. This is a NCBI updated version of how DNA sequence depository of NCBI has been increasing over the year, and how the cost of per megabase DNA sequence has decreased so significantly. Now it's so cheap to assemble a sequencer organism. So let's go further on what a genome sequencing project takes to put together a genome of an organism. So what you do, like you decide organism which you are interested in and extract DNA. And DNA extraction is one of the very, very crucial part of it. It has become more crucial with the long-read sequencing technology, but it's not that important for short-read sequencing, but still it's a very, very critical part of it. Many complex organisms have very difficult to extract DNA, high quality DNA, and that create a lot of trouble in producing good quality sequence. So you have good DNA, then what you do, like you sequence it, the technology at your disposal. Like Illumina, you're doing Illumina, what is the purpose of it? Whether you want to just variant call, then Illumina would suffice. But if you have to go for Genome Assembly, then you have to take help of some other sequencing technologies such as PacBio. So you do a quality filtration of the data, initial data. Then again, when you do sequencing, you have to put adapter to a sequence which need to be trimmed before you go for the sequencing assembly process. Then you assemble and then do assembly validations. We'll go with one by one. I try to explain the simple concept rather than going into complexity of the genome assembly. So DNA, as I discussed in last slide, the DNA is very, very crucial part of it. And I have, during my PhD, I was trying to sequence genome of rust pathogen. Rust are a group of fungi which infect wheat and cause very devastating disease like wheat yellow rust and stem rust. So as the pathogen is a biotropic in nature, so you can't grow in lab. The only tissue you have is spores, which are very, very complex tissue, full of nucleic lipids and complex lipid, which make it very difficult to extract very high quality DNA. And I did a lot of optimization in order to extract high quality DNA that can be run on nanopore sequencing technology. I'll discuss a little bit about how wonderful this nanopore sequencing is, if time permits. So there are many contaminants you could have. Like one is most likely contaminant are inherent to the tissue you're using. Other than like in your extraction protocols, also you can end up having a lot of phenols. You use organic compound like phenols, and they can also end up contaminating DNA. So you can also share up DNA by ubiquitous pipetting and vortexing. So you have to take care of all these things depending upon what is your aim is, like whether you're going for a long read sequencing, then these things become really, really critical. Like if you aggressively, you end up sharing all of the DNA. Then some of these organisms, like these are say insect cells are made of chitin, which are very, very tough. And how do you extract good quality DNA like from these? So that become then again these mushroom. Then I discuss about the rust fungi, and there are plant like conifers and redwood tree, which is very, very have a lot of only compound. Then once you have DNA, you'd have to determine whether this is pure or not. So the purity of DNA is defined by two ratios. You might have read in your theoretical classes and practical that DNA purity is defined at the level of 260 by 280, where 260 is the absorption of the nucleic acid, and 280 where the absorption of protein, like histidine is the one which absorbs the UV light at 280 nanometer at the most. If you have this ratio 2.8 to 2.2, then anything less than 2 indicate that you have some phenol protein contamination, and anything beyond to say that you have a lot of RNA in your sample. Then another way of determining purity of DNA is the 260 by 230 ratio. 260 again is where the DNA absorbs UV light, and 230 is where many organic compounds which contaminate like polyphenol, urea, guanidine, they absorb light, and if they are in your DNA preparation, then they're inflating or deflating the reading, which it's supposed to be 2, and if you deviate from 2.2, then you expect this kind of contamination. If you don't do this QC state, you might end up getting poor quality results of your sequencing there. So then again, for long read sequencing, it becomes crucial whether your DNA is shared or intact. So long read sequencing technology, what happens when you put high quality long read DNA, those read translate into the sequences. So for example, nanopore, nanopore is where like your DNA molecule goes through a single pore, and the longer your DNA, the sequencing technology is such, it does not have any limitation on the read length. It's only the matter that longer the DNA sequence you would have, longer the read you're going to get. So you have to make sure that you have really high quality DNA, which should not have this kind of smear. If you have this, you end up having this kind of smear, you're more likely to get very poor quality DNA, your read length, which again defeat the purpose of long read sequencing. Long read sequencing means your aim is to produce really long read. And if you have shared DNA, it's not going to get anywhere from there. So these are the histogram, just to show like if you have like, so this, see the image below, the band looks very intact, and those bands translate into the very high read length. This is read length on the x-axis. And if you have the shared DNA, then you probably have the spread of the reads, because you have a lot of short read data also. So yeah, and for a long read sequencing point of view, you have to make sure that you have DNAs of highest quality. So once you have DNA, then you have to assemble it together. You have to put DNA sequences one after other in order to produce a contiguous sequence. So there are multiple ways you could do this. One is the clone by clone approach, which is the classical approach, no longer being used these days. Mostly it is the whole genome shotgun sequencing, where you just fragment your DNA, sequence it, and then try to use the complementary DNA complementarity as a basis to produce the whole chromosome. So in clone by clone, what you do, just to briefly mention it, what happens into it. So you fragment your DNA in a really large chunk, and then clone into vector artificial chromosomes, which is called bake, and then you try to sequence one clone at a time. So the way is try to reduce the complexity. If you try to assemble all of the genome in one go, it's very, very complex. So what you do, just take one bake clone and fragment it and try to assemble it one by one, then take another bake, then you so it's take a lot of time, and it's very expensive also. So but it's very, very precise one. That is the advantage it has. Anyway, let's go to this is again how the clone by clone sequencing work. You produce one bake or react sequence, then try to put them one after another using your mate pair sequencing, using either false bit or East artificial chromosomes. So So bake by bake, clone by clone approach is no longer being used, but the assembly technology is being used is the shotgun sequence technology, where you just fragment your DNA, then take the help of base complementary using assembly algorithm. There are many assembly algorithms which is out of scope of this today's talk, but there are that's mathematical way to devise algorithm in order to put the assembly together, DNA read together. So, but what happens for in a simplest way? I'm trying to represent a cartoon where try to show how you put together sequence after another using base complementary. So you have this 70 base pair stage. Then you have another different reads. When you do sequencing, you get a lot of reads, and on just basis of complementary, you see this 70 base pair has complementary to these two reads, because on the basis of base to base complement. And on the basis of that, like you have started with 70 base pair, now you have almost like more than 50 base pair read just on the basis of complementary. This is how the shorted what you call shotgun assembly and sequencing works. This becomes 66 base pair just based on the complementary. So another important concept to discuss here is the coverage. So how many times you have to sequence a base pair in order to decipher whether this is a G here or there could be something else. So all of the sequence if you have G, then you have like 100% confidence. This identity is 100%. So you are very sure that this particular base is G. But again, here is of coverage is again very high. So you would here this C indicates an error in the sequencing. So you could easily decipher that this most likely base pair on this position is A, but not the C. But how about this? You have just 50% confidence here. If this base here either could be G or C. So this is not a sufficient depth of data you have. So in order for this particular position, you need more information in order to ascertain what base sequencing here. So that's where come the concept of the depth of coverage and coverage of your data. So more the data you have, more coverage you have, more confidence you're going to have on your assembly. So once you have done the assembly, you could visualize. There are tools like IGV, one of the tool where you could see like whether how your assembly look. But it just you could only see in the windows. Genome assembly size can be in MBs. So it was no use, but generally, if you like to see how actually the assembly looks, then one of the way to look into. So there are challenges with the genome assembly, and I'm not going to go into a lot of detail about these because these are can be very complex. So these three major challenges associated with the genome assembly are the heterozygosity. Heterozygosity is what heterozygosity means like once if you're dealing with a diploid organism, it has one chromosome copy from maternal, one chromosome copy from paternal, and they have their own identity. Though they call homologous, but they have their own identity and they have their own. So that cause heterozygosity. Another way of heterozygosity, especially in the clonal plant organism, say for example potato, rust, and many other organisms which generally reproduce asexually. When you reproduce asexually, you accumulate a lot of mutation because there's no sexual reproductions and there's no purifying selection happening. Because once you have a sexual reproduction, you reshuffle your DNA and then you have a purifying selection. That like take care of the lot of heterozygosity acquired during the lifespan of an organism. But once you reproduce asexually for a very long time, you only reproducing through mitosis, going through meiosis. and there's no verifying selection, then you for the hundreds of years, some of the organisms like potatoes, sugarcane has been grown and multiplied by asexual means. So they end up getting very heterozygous because both copy accumulating DNA mutation independent of another copy. And you say this was the starting point after your sexual reproduction, as sexual reproduction. But once you go through a cycle of asexual reproduction, you having their own, the chromosome nuclei have their own identity, that they become on their own. And if this keep happening, then you have achieve a level of heterozygosity, which causes a lot of complexity when you assemble the DNA electron. So this, for example, this is the diploid organism, but you have these homologous gene regions. This light blue and dark blue are homologous, but you see this part of the genome is heterozygous. If you try to assemble this these heterozygous region with a short read, what you are going to end up, either you can assemble either the red fragment or green fragment, and hence, but long read, see the size of the long read. Short it not able to span these repeat region, this heterozygous region. So this is heterozygous region is this big, and their short read are this big. How it's going to know where this particular read either coming from the red region or green region? You could not understand this. But once you have long read, really long read, that can span from whole of the heterozygous region, then you would know whether it's one of this read belongs by the red or green. And this long read, this is how long read help in assembling better genome. See, highly heterozygous genome region tend to assemble separately. That only happens with the long read sequencing. To tell you the thing, with the short read sequencing, it try to collapse the assembly, and then you're losing a lot of information. Homologous region exists in multiple copies in assembly. Yeah, homologous region generally the chromosome are homologous, so you expect like 80 to 90% of the genomic region are homologous to each other. So this is one of the graph to try to show you how heterozygosity affect the assembly size. So if you reduce the heterozygosity, the assembly size, if you have a very high level of heterozygosity, so your assembly size would be, let's say you have 100% heterozygosity. Loss of heterozygosity, 100% loss of heterozygosity means like your genome size would be collapsed. You have only single copy. You won't have, if you have very high level of heterozygosity, your genome size would become because that the genome going to get phased into two copy, maternal and paternal, because they are very, very diverse from each. And the another challenge associated with the genome sequencing is the repeat. Repeat are the kind of menace when it comes to the genome sequencing, because repeats are everywhere, especially in the complex eukaryotic organism. So, for example, human genome is 3 billion base pairs, but only 3% of it code for the protein coding sequence genic region. Genic region generally balanced with the ATGC content, like you have 50-50% base pair compositions. But when it comes to the repeat region, which can be highly AT rich or can be highly GC rich, and you have homopolymers running like A, G, and for a long stretch, which create a lot of complexity in assembly. So this in situ hybridization is showing this identical sequence, exactly identical sequence of can happens in multiple positions in the chromosomes. It's just one of the way to see the repeat region in the chromosomes. They cause assembly breakers. Very often you get very fragmented assembly because of the repeats, because repeat could not be resolved small read sequencing such as Illumina, but that has been largely taken care of by long read sequencing. but still there are very complex regions which cannot be can't be dealt with the long read sequencing. So for again, you have to, some technological innovation has been happening, which I'm not going to discuss, what we call context genomics, and many other technology has come into our life to save our life. So I just don't want to go into this is again just try to show you how repeats can affect the genome contiguity and affect N50. And on the contrary, long read sequencing, how wonderfully can deal with the small repeat regions. Another cause of concern for the genome assembly is the ploidy. Just to give an example of the ploidy, this is a wheat genome sequence. So wheat genome sequence, how a wheat genome sequence came to be, what wheat we eat today, like Triticum stivum, is a composition of 3 different genomes. So one genome comes from different species called Triticum monococcum, and these 2 other copies come from Azulopsis, different species Azulopsis, and then that make together a genome of. So when you sequence wheat genome, you just can't imagine the level of complexity you're dealing with. Like you're not going to assemble one genome, you have to assemble 3 genomes altogether in order to decipher the complexity of this genome. So, and many plant organisms, like in animals, ploidy is not a problem. Mostly, most animals are diploid. But when it comes to plant, like it's really hard. Wheat is the one example. Then sugarcane, potato, and mustard, many groups. So once you have genome, then what is the next step is to what kind of genes, how many genes are there, what those genes code for, whether they are transcription factor, whether they are like single copy genes, multi-copy genes, what agronomic agriculture trait they're affecting. So this is what the next step. Again, very heavily depends on bioinformatics using different approaches, ab initio. So these are the different approach for genome assembly. This is ab initio is likelihood-based method. We use a lot of information already available in public domain. Then hybrid approach where like ab initio as well as the genome features defined based on the pattern across the genes in the chromosomes. And there are many other ways, like you also use the transcriptome data of an organism. So when you try to assemble, annotate a genome, you generally also sequence transcriptome of that organism, because the transcriptome is the express part of the genome. So what are the genes are there. So once you also sequence transcriptome of that organism, that significantly help in putting together annotation of that organism. So these are the standard features, for the understanding of the master student, like how a gene is defined. So you have DNA, then pre-mRNA, mRNA. So mRNA have certain features, like it has a UTR, then you have a initiation codon, then termination codon. So what you call defined in the ORF, operating frame. So these are the features generally used in the ab initio model of gene prediction. So this is what I wanted to discuss. I don't know. This is during my PhD I used this nanopore, very small genome sequencing instrument. It's just like the size of a pen drive, but it is a very useful sequencing technology which has been very massively used around the world to genome sequence eukaryotic complex organism, because it produce really long read. And it's just a pore-based sequencing technology where like you have designed a nanopore. When DNA passes through that nanopore, there's a current disturbances. The membrane have a current disturbances. Those current disturbances has been then using neural network, help of neural network, machine learning. You decipher what are the bases passing through that membrane, nanopore, and you could have the nucleotide sequence just passing DNA through a single pore. So what are the take-home message? Eukaryotic genome are complex. Yeah, they are really, really complex, and small reads are not able to deal with this such complexity. Longer reads definitely help in assembling highly contiguous genome. And DNA quality is paramount for long read sequencing technology such as Nanophore. And this is some outreach. So our institute has been pioneer in sequencing many genome with the help of international consortium. So we have sequenced rice genome with the international consortium, including many 16, 17 countries. Then we also were part of sequencing tomato genome sequence. And we have been also part of wheat genome sequence, which came recently last 2 years. Thank you everyone for your attention.
Thank you, sir. Thank you, Dr. Ramavtar. Indeed, it was a very, I would say, a wonderful session, and thank you so very much for enlightening our participants with the detailed information about genome assembly and the annotation. It was like explain, you tried to explain each and every aspect in this short span of time. So we are highly grateful to you for your wonderful presentation, and you have shared very the valuable facts about this genome sequencing. Thank you so very much. Now I invite our Dr. Preeti Rawat to propose the vote of thanks to Dr. Ramavtar. Dr. Preeti Rawat.
Thank you so much, ma'am. Good afternoon. I think it's good morning. Good morning, everyone. Myself, Dr. Preeti Sharma Rawat, Associate Professor in the Department of Life Sciences, Radboud University. And on behalf of the university, it is my pleasure to convey this vote of thanks to our Honorable Speaker, Dr. Ramavtar Patyalji. Thank you, sir. Thank you very much, sir, for enlightening us about very so simply about what the genome is, its sequencing and assembly of the genomes after the sequencing, sir. And I'm very much sure, sir, that our students and the faculty as well, including myself, have gained knowledge about some popular and common techniques of their step-by-step mechanisms for the genome sequencing and the requirements for the genome sequencing, and of course the limitations that are there in these techniques that we use for genome sequencing, and also the challenges involved within the same. So, sir, thank you very much once again, sir. And now I invite Dr. Simarjit Kaur Shergill, Head and Department of Life Sciences, Dean Students Welfare Association, to carry forward the session and convey the concluding remarks. Please, ma'am. Thank you, everyone, once again.
Thank you, Dr. Preeti Rawat, and thank you so much, Dr. Ramavtar, for enriching our knowledge and the understanding in the gene sequencing. It was well needed for us, and I am thankful to Dr. Patel to get you over here on this platform. So thank you so very much.
Thank you.
So a very good morning once again to all of you. Now I would like to give the concluding remarks about our 3 days workshop on life science, food science, and environmental technology. So our first day observed Dr. Lakhvinder Singh, who spoke about the wastewater treatment techniques. Dr. Anu Agarwal, she spoke about the various food processing techniques. Dr. Simranjit Singh, who spoke about the constructed wetlands. So the first day was very, very much productive and the fruitful for our participants. So likewise, we moved forward for the second day of the workshop. And the second day of the workshop observed Dr. Bindu Rani. She spoke about the screening and the analysis in the genetic diseases. Dr. Archana Chaudhary enlightened our participants about the stem cell technology, its applications and the ethical concerns. And Dr. Simarjit Shergill spoke about the air pollution and its disastrous impact on the human health and the environment. And the third day of the workshop concluded with the sessions of Dr. Sonia, who enlightened our participants about the gene sequencing techniques. Dr. Ramavtar gave a detailed information about the genome sequencing, assembly, and the annotation. So we see what a wonderful session we had during these 3 days of the workshop. And I'm highly, highly grateful to all the speakers of all the sessions for sharing their expertise with our participants and enhancing their knowledge and understanding in the diverse area of their own specialization. I'm highly thankful to our honorable management of Ratan Bhara University, were the Vice Chancellor of Ratan Bhara University, Professor Dr. Parvinder Singh, respected Registrar, Professor B.S. Satyal, Deputy Registrar, Deans, heads of the department, faculty members, especially from the Department of the Life Sciences, for their support and the cooperation for the successful execution of this 3 days workshop. And of course, this workshop could not have been successful without the active participation of our students. So I really owe a big, big thank you to all my dear students and the participants. So with all these greetings, I now would like to close this 3 days workshop, and I, Professor Dr. Simajit Shergill, sign off from this workshop. But yes, we will be meeting soon in personal or through any virtual platform with another round of the workshop. So till then, stay safe and follow all the COVID-19 protocols. God bless you all. Have a great day ahead. We will be meeting soon. Thank you very much, everyone. Thank you.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, ma'am.
Thank you, everybody. Thank you.
Thank you, ma'am, once again.
Thank you. Thank you, Dr. Archana, for joining us. I can see you.